Hello Biology 107 students. Today we're going to talk about translation. That's where we're going to go from an RNA sequence and we're going to build an amino acid sequence. This all happens at the ribosome. Hey, here's a nice picture of a ribosome. We're going to talk lots about these ribosomes. So, back to this, the central dogma of molecular biology. First we have replication. We've already covered that. Then we talked about transcription. Today we're going to talk about translation. These are super important. Make sure you put a big star beside these notes. This is what the final exam is going to focus on. 100%, this is a promise, not a hint. Okay, so before you dive into translation, why don't you go check out this video. I have it linked in the PowerPoint. I also have it linked with the course playlist. It's a great video. It's put together by our textbook publisher, and it covers a number of the things we've talked about, and it's also going to talk about translation. Go ahead, go do that right now. Okay, so hopefully that gave you a pretty good overview of that video. It's very well put together. Now we're going to dive into the details on translation. So first let's talk about the components. First component is messenger RNA. There it is, over there in red. So we talked about how we're going to make the messenger RNA through the process of transcription. Secondly, the amino acid transfer RNAs. So those are the transfer RNAs, and they have amino acids attached to them. Notice it says amino acyl. So acyl, the YL infers it's a group attached to something. So normally this would say amino acid. But acyl means the amino acid is no longer an acid, but it's actually a derivative, so that YL. So when we say amino acyl transfer RNA, it means we've attached that amino acid to a transfer RNA molecule. Third thing is the ribosome. Yay, ribosome. Notice the ribosome has two subunits. It has a large subunit here and has a small subunit here. So let's talk about these things. So first, here's the transfer RNA. So notice the transfer RNA, like all nucleotides, has a 3' end and a 5' end. The 3' end, this is where the amino acids are going to be attached. So we'll talk about that in a moment. Down here we have that anticodon. This is what is going to recognize a sequence on the messenger RNA. This is the adapter. This is the translator. This is how we decide which codon corresponds to which amino acid. This is a two-dimensional structure. Here's what a three-dimensional structure of transfer RNA looks like. It actually has, unlike most other RNA molecules, some double-stranded regions. You can see all the nucleotides are coded there on the left. This is a little bit hard to draw every time, so the textbook is going to use this thing here to represent what the transfer RNA looks like. Notice we have the anticodon. That's important. We have the amino acid attachment site. That's what's really important, those two things. So something important to know about these transfer RNAs is we don't just have one of them, we have 20 amino acids. So we have more than 20 amino acid transfer RNAs. Not exactly 64, some of them double up on different codons, that's not really that important. But something that is important is that we have to get the amino acids onto these things. So there's the transfer RNA. We take these enzymes here. These are called amino acyl transfer RNA synthetases. So that's the name of the enzyme that is going to attach this amino acid. So we have alanine here. We're going to attach it onto this transfer RNA. Here goes. Boom. There it is. So this is alanine. There's the anticodon down here. We're all ready to go. This, of course, takes energy. We're going to use energy in the form of ATP. Quick question for you. Some true or false to test your knowledge. True or false, ribosomes are made of ribosomal RNA and proteins. True. Ribosomes are made in the nucleus. True. If you want to be a bit more specific about this, ribosomes are actually made at the nucleolus. Remember the nucleolus was that dark patch in the nucleus? So what's going on in the nucleolus? A few things, it turns out. We're making RNA there, that's why they look dark. 2, ribosome proteins are coming in and assembling with that RNA. The ribosomal proteins actually are made in the cytoplasm by other ribosomes. All these things put together and you get the subunit. So the small subunit and the large subunit, they're all put together at the nucleolus. Number 3, ribosomes can be cytosolic or bound to the smooth ER. If you guess true, you're wrong. What's wrong about this? Well, notice the word smooth. Almost got you there. The true answer would be the rough ER. So make sure you read all of your questions carefully. Very important, read your questions carefully. So here are the ribosomes. There's the subunits from a couple of different bacterium. The structures for these subunits aren't determined on every organism on the planet, so we have, it looks like a large subunit from this organism here, Haloarcula, and the small subunit from this Thermus thermophilus organism. And let's take a look at the ribosome in a bit more detail. Ribosome is made of RNA, that's all that brown stuff. It's actually more RNA than protein. The protein is the blue portion, and is actually the RNA that has the catalytic activity. So just a reminder that we have different types of ribosomes. Prokaryotic ribosomes are smaller. They're 70S. They're made of a 50S large subunit and a 30S small subunit. The eukaryotic ribosomes are 80S, made out of a 60S subunit and a 40S small subunit. So closer look at the ribosome. Here's a model of the ribosome. Notice it has 3 binding sites here. They're called the A site, the P site, and the E site. So hey, that's kind of cool. It spells a word for us. So that's useful. We can spell out the word ape. And actually, when we talk about translation, we're going to go in order. We're going to go to the A site, to the P site, and then to the E site. So what are these sites? Take a look in the top right. A for amino acid, or if you want to be really technical, it's amino acyl transfer RNA. That's what's going to dock in that site. P is for peptide bond. If you want to be technical again, peptidyl transfer RNA. So we have a peptide bond attached to transfer RNA there. We'll kind of get into some specifics in a couple of minutes. E is for exit. So we have 1 for amino acid, 2 for peptide bond, and 3 for exit. And that is the order of the things in translation. Let's take a closer look. First step of translation is initiation. This is where things begin. So notice all these things that have to come together. We have the small ribosomal subunit. We have the messenger RNA. And what's not shown on here, or actually it's shown up a little bit higher, is the messenger RNA has a start codon. So there it is, AUG. So that is lined up, and this is actually lined up right at the P site. So I'm going to put a P there to remind us that this is lined up at the P site. Last thing that comes in, we have this initiator transfer RNA, and the initiator transfer RNA has methionine attached to it. So notice the anticodon, I'll just say anti, it's hard to write with my mouse. The anticodon is lined up with the start codon. So the start codon is AUG, the anticodon is 3' UAC or 5' CAU, depending which way you want to read it. So once those are together, we need a little bit of energy in the form of GTP. So remember, we like to use ATP a lot. Well, it turns out GTP has exact the same amount of energy as ATP, and some processes actually prefer GTP over ATP. This is one of those. It makes GDP and inorganic phosphate. That energy comes in, and basically it allows the large subunit to dock, and everything is ready to go. So like I said, notice here that at the P site we have the initiator transfer RNA, and we have our first amino acid shown right here. So this has a name, it's called the translation initiation complex. Okay, let's go to the next step. Okay, so let's take a look at the next step. The next step is elongation. So in elongation, remember those steps. We've got A, P, and E. We're going to start with A. A of ape. A for amino acid. So we're going to bring in this new amino acid. Here it is, the new amino acid. So amino acid A. And the new transfer RNA. So T, R, N, A. There we go. And then down here we have an anticodon. So that anticodon is going to match with the next codon that is found here at the A site. So it's going to be complementary and anti-parallel. So we have this codon codes for the amino acid that's found on this transfer RNA. So here it goes. It's going to dock there in the A site. There we go. So step 2. Notice, by the way, each of these steps does take a lot of energy, and we have our GTP right here getting converted to GDP, an inorganic phosphate. So step 2 is the P part, so P for peptide bond. So notice here we have this peptide. So here's the peptide, okay? And it has 4 amino acids, and this peptide is going to get transferred right here. This bond gets transferred over here to this other amino acid. So now we're going to have 4 in a row, and if you take a look, now we have 5 amino acids in a row. The new peptide strand is now transferred to the A site, but we have 1 more amino acid, so this is good. Now we have a slightly longer peptide chain. Let's go to the next step. So the next step, again, takes some GTP. Like I said, we have this amino acid strand of 5 amino acids found here at the A site. The next step is something called translocation, where everything just shifts over a little bit. So if you take a look, this particular strand that was at the A site is now here. You can see it is at the P site. So that's good. Now we have a strand of 5 amino acids at the P site. This old transfer RNA ends up in the E site. E is for exit. It doesn't bind there, it just slips out. It's shown the door, it's exiting. So after this, notice we've done one entire cycle, except for now we have an extra amino acid. We can proceed on to the next step, which is going to be a new transfer RNA is going to come in. It's going to recognize the new codon. It's going to deliver a new amino acid, and this is going to rinse and recycle. So it's going to go around and around and around. Typical protein has maybe 100 to 200 amino acids, so this cycle will happen 100, 200, sometimes even more times. The last step, termination. That means we're going to stop the whole thing. So take a look here. The next step, we have a codon, a stop codon, UAG, UAA, or UGA. So if that comes in, there is no transfer RNA for those particular codons. They're stop codons. Instead, we get this thing here, a release factor. A release factor is basically a protein, and a protein that comes in and recognizes one of those codons, those stop codons, and basically disassembles the entire apparatus. So here goes. You can see the old transfer RNA is released, the polypeptide here is released, and lastly, the subunits and the messenger RNA, all of these things are released. And that is how translation goes and how it finishes with the process of termination. So take a break here again and take a look at another video. This one is relatively short, it's just 2.5 minutes long. The resolution isn't super high, the quality isn't super high, but it is actually a very good explanation of the whole process of protein synthesis that happens at the ribosome. So go check out this video now. It's found at the playlist for the course, or you can find the link in the PowerPoint if you download it. Go check it out. So here's another slide just from the textbook showing the processes of transcription, So starting here at the top, transcription, we're making our messenger RNA. Notice it has some processing going on. So remember, we have the capping and the tailing and the introns getting spliced out. It's leaving here through the nuclear pore. Notice it now has a poly A tail and a 5' cap. The 5' cap allows it to find a ribosome. It docks at the ribosome, and you can see all the processes of translation happening, and we're making a protein. So as the protein is formed, as those amino acids are strung together, it starts to fold. So we have hydrogen bonds forming, and you have other intermolecular forces such as ionic charges and hydrophobic forces, and the protein starts to form a structure. You can see I have it making some alpha helices there. And so one reminder is that this happens in two places. Some proteins are made in the cytoplasm. They're made with free ribosomes, and these are usually soluble or cytoplasmic proteins. Other proteins are made at the rough endoplasmic reticulum. So these are made by what we call as bound ribosomes. They may also get glycosylated. That means we're adding a carbohydrate, and that can happen both at the ER and the Golgi apparatus. These proteins here are usually targeted to specific locations in the endomembrane system. So they might go back to the nuclear membrane, they might go to a lysosome, they might go to the plasma membrane. Last thing to keep in mind is that many proteins may have other things done to them. We're going to cover this briefly in our next unit. Some of the things that happen after translation, so post-translational modifications. So this is the end of part A. I wanted to break up these videos into relatively short digestible units of 15 to 20 minutes each. So I hope you learned something in part A. Part B will mostly be on mutations that can happen during translation. I hope everyone's doing well. Talk to you later.